In this video, we're going to look at application questions that are about cell structure. At the end of this session, you will be able to define different command words, learn how to read A-level exam questions, and confidently answer application questions. AQA uses lots of command words in their exams. The full list can be found here. If you want to make a note of that, pause the video and you can have a look now. However, I've picked a few for you to look at. These are the ones that are most common to come up in your biology exam. On the next slide, have a look at the command words and check you understand what they mean. So here are the most common command words that I have seen in your biology exams. Take some time now, pause the video to check that you understand what they mean, write down their definitions, and then you can check them with the next slide. Okay, so here are the answers. So, annotate. So you may be asked to add a notation or a label to a graph, diagram, or other drawing. Apply is when you take your knowledge and you apply it to perhaps a situation you've not heard of before, but it is knowledge that you have learned as part of your A-level specification. Calculate is to work out the value of something. Compare is to identify similarities or differences. Define is to specify a meaning of. Describe is to give an account of or a very basic overview. Evaluate is to judge from available evidence, so that could be a data question. Explain is to tell me why, give me reasons as to why something's happened or happening. Identify, so name or otherwise characterize. Predict is to state an outcome, a plausible outcome using knowledge you already have. And suggest is to present a possible case, but I want to go more into the suggest questions. So today we're going to look at application to apply questions, but we will look at some suggest questions and some other types of questions as well using these command words. So, application questions. Why do exam boards make you not only learn a load of information, but to apply that information too? It's because they don't want to know that you've learned every example in the book by heart. They want you to take that knowledge and apply it to different scenarios, as ultimately, in any scientific career, this is what you will be doing. However, it's a really difficult skill and it causes a lot of controversy. If you're on any social media, you will see at this time of year in exams, you will have a lot of controversy. So, for example, seals weren't on the OCR specification in 2010, but they still asked a question about them. Also, we did the practical about osmosis in potatoes, but then they asked you about carrots. But remember, carrots are still made up of plant cells, so you know the answer to that question. So how do we deal with these application questions? Firstly, be interested in biology. Read widely around your subject. If you already know what a Weddell seal is and it lives in Antarctica and has hemoglobin with 1.6 times more oxygen carrying capacity than human blood, you're not going to be as thrown by a seemingly random question on seals, because you'll already kind of have an idea of what it's going to ask you about. 2, I cannot state this enough, practice, practice, practice. The more application questions you do, the more you'll be used to them. But do not just read them, think that's hard, look at the mark scheme or copy the mark scheme. That is not going to work. Application questions are there to make you think. So you need to think. So make sure you do sit for 20 minutes or go away, make a cup of tea, still thinking about it. And eventually you should come up with some knowledge that you have learned in class that you already know to answer that question. Then have a go. Just put something down. Do not worry if it's wrong. Okay? It's that thinking process you need to get used to. When you first start this, you will not get every question right. But you need to get used to this process. Then check it with the mark scheme. If it's wrong, make sure you understand why it is wrong. So correct it, but make sure you understand why you got it wrong. If you do not know this, look it up, speak to some other students, speak to your teacher. You need to understand why you got it wrong. Then a couple of weeks later, check, do it again, see how much better you do. And thirdly, be confident. You have the knowledge to answer every question on your A-level paper if you have done everything that your teacher has told you to do. You will have done something similar in class. They're just going to ask you something like cell structure in crocodiles rather than in humans. But you know about cell structure, you know about animal cell structure, and you smash that task in class. So get that answer down, be confident. Okay? So let's have a go at some application questions. So these are real life examples of past paper application questions. So first one we're going to look at is about stomach ulcers. And stomach ulcers are caused by damage to the cells of the stomach lining. People with stomach ulcers often have a bacteria, Helicobacter pylori, in their stomachs. A group of scientists is interested in determining how the infection of H. pylori results in the formation of stomach ulcers. So they grew different strains of H. pylori in liquid culture, and the table below shows the substances released by each of these strains. So you can see that strain A releases a toxin and an enzyme that neutralizes the acid. Strain B only releases the enzyme, and strain C only releases the toxin. Okay, so it's asking about H. pylori, which you might not have heard about before, but you've heard of bacteria. So you do have knowledge to answer this question. The scientists then centrifuged the cultures of each strain to obtain cell-free liquids. You've done centrifugation, so you should be able to answer this question. Even though you've not really heard about centrifuging to make cell-free liquids, you know how a centrifuge works and what the basic principles are. They added each liquid to a culture of human cells. You know about human cells, you've learned about those. And then they recorded the amount of damage to human cells. And the results are shown below. And the error bars show plus or minus 1 standard deviation. So you can see the standard deviations there as well. So you might not have heard of H. pylori, but as I've just said as we've gone through the question, you've heard about quite a lot of stuff in this question. You know about bacteria, you know about centrifuges, you know about human cells. So you do have knowledge to answer this question, even though it might have seemed a bit weird when you first opened the paper. So they're going to ask you questions about this data. So the first one is to describe and explain how centrifuging the culture allowed the scientists to obtain a cell-free liquid. So you need to describe, so state what you're going to do, how you're going to do that, but then explain why you were doing it as well. You will get no marks if you do not add explanation in. You need to both state what you're doing, describe it, and tell me why you're doing that. Okay, pause the video and have a go at the question. Okay, so the answer is, first of all, that the cells are large, dense, and heavy. That's explaining why this works. So you need to say for the first point, large, dense, heavy cells. The second point is saying when you centrifuge it, they move to the bottom of the tube. So that forms a pellet. And the third point is then saying that liquid on top, the supernatant, can then be removed. But you must refer to whole cells. I'm going to show you the examiner's report now so you can see where students go wrong. Firstly, students will write all they know about the use of a centrifuge in cell fractionation. That's because this is what you learnt about in class. But we're not talking about cell fractionation. We're not splitting the nucleus from the mitochondria or the ribosome from the lysosome. Rather, we are applying our knowledge to the use of a centrifuge to obtain a cell-free liquid. Mark points 2 and 3 were still awarded if you got the first one wrong. So if you talked about cells being homogenized, you couldn't get the first mark because you've not talked about whole cells, but you could get the second and third because you were talking about the centrifuge. However, mark point 3 was rarely rewarded as students referred to removing the pellet. You do not remove a pellet, you remove a liquid on top, which is the supernatant. If you removed the pellet, it would break up and you'd mix the contents once again, and that wouldn't be very helpful. So just be aware of where students go wrong. Okay, so in case you've forgotten what the question is about, you can pause video and have a look now. Okay, so the next one, the scientists measured cell damage by measuring the activity of lysosomes. Give one function of lysosomes. And remember to state the function. Don't tell me what a lysosome is. Don't describe a lysosome. Give one function of a lysosome. What does it do? Pause video and have a go. Okay, so lysosome, its function is to break down cells, cell parts, or toxins. See the idea of breakdown or digestion. Students were really good at recalling the function of lysosome, but where you could fall down on this, and this is why I said do not describe what a lysosome is some only stated that lysosomes stored enzymes rather than identifying their active role. Okay, so just in case you want to re-go over the data, have a look at this. Pause the video and you can carry on in your own time. So the next question is, H. pylori cells produce an enzyme that neutralizes acid. Suggest one advantage to H. pylori of producing this enzyme. Now, suggest questions, most students find really difficult because it's almost too open. The word suggest means you can put pretty much anything. However, it isn't very open at all. And you only have a very limited number of marks you can still get for this. So what I would suggest that you do is get rid of the word suggest, and it tells you exactly what the examiner wants. So get rid of that word suggest, and it tells you they want one advantage to the H. pylori of producing this enzyme. So just state one advantage to the H. pylori of producing this enzyme. But remember, the question is 2 marks. So give the advantage, and then perhaps say why that might be an advantage, because it is 2 marks. So the advantage needs to have 2 points to it. Okay, pause video, have a go. So here are your 2 marks. So the first mark is to stop them or reduce them being damaged, destroyed, or killed. You cannot say denatured because it is not an enzyme. Protein is denatured, bacteria is destroyed or killed, an organism is destroyed or killed. Now the second mark, and it must be in the context of the stomach, is to say by stomach acid. So you need to say what the advantage is and why that's an advantage, because the stomach acid there. Okay, so think about the 2 marks. Always look how many marks there are and make sure you include lots of relevant information. Relevant is the word there. Okay, so where do students go wrong? Well, it's generally good, but bacteria being denatured, it is not a protein. Proteins are denatured, it is killed or destroyed, or neutralizing acid would lower the pH. Remember, your pH scale gets higher the less acidic something is. Okay, so the wrong way around. So make sure you're happy with pH scales and things like that. Very basic science knowledge is always good to have. Okay, now for the next question, you do need to look at the data. So if you've forgotten, have a look. I will give you a little bit more in a moment. So D, what do these data suggest about the damage caused to human cells by the toxin and by the enzyme that neutralizes acid? And explain your answer. So it's about the damage caused to human cells by the toxin and by the enzyme, but it's asking about the data. So you need to use the data to explain your answer. Now again, it has the word suggest in it. So again, remember, suggest does not mean give your personal thoughts and views, but it does suggest that it might. Instead, it wants a fact. So a more accurate question might be, what does the data show about the damage caused to human cells by the toxin and by the enzyme that neutralizes the acid? So tell me what does it show? And have a look. So it's asking about the data. So you've got data there. You need to look at the graph. Tell me, is it worse if you've got the toxin and the enzyme? Is it worse if you've just got the toxin? Does it make relatively no difference if you've just got the toxin or just the enzyme? So think about those kind of things. So pause video, use that data to answer that question, and then have a look at the answer. So here is the answer. There is more cell damage when both are present. So in type A, you get more cell damage. However, you do get some cell damage when either they're on their own. So some cell damage in B and some cell damage in C. And you must say B and C. If it's underlined, you have to say that word. So you've described there what is showing you. But you then need to use the standard deviation. So there's error bars. If error bars do not overlap, it means the difference is real. So there's a significant difference. If error bars do not overlap, it means that the difference is not real. So there's no significant difference. In this graph, standard deviation does not overlap for A with B and C. So A doesn't overlap with B and A doesn't overlap with C. So therefore, the difference between A and B and A and C is real. So you must say and so in that answer, otherwise you will not get that point. Standard deviations, however, do overlap between B and C, so there's no real difference there. For mark points 3 and 4, both aspects need to gain a mark. So you need to say which ones overlap with each other, and then you need to say whether the difference is real or not. Okay? So mark points 1 and 2 were commonly awarded but very few students could actually describe the relevance of standard deviation and overlap. So some students tried to explain why the damage was caused to human cells. It's not asking you that, it's asking you to explain what the data shows. So make sure you read the question. Okay, so that was a question about Helicobacter pylori with some data analysis. Today, for the rest of this, we're going to have a look at a comprehension question. So for comprehension questions, you will be given a paragraph to read. So I want you to pause the video and read through that paragraph now. So they'll take that paragraph and they will ask you some questions about that paragraph. So the first one is to explain the link between mitochondria and muscle cells, and it's referring you to lines 6 and 7. So make sure you read lines 6 and 7 first before you have a go at this question and explain that link. Tell me why there is a link and what the link is between mitochondria and muscle cells. Okay. So there's 3 marks this question, so you need to think about 3 points. First of all, mitochondria is the site of respiration. I will put aerobic respiration because sometimes they want you to put the word aerobic. Therefore, mitochondria is the site of production of ATP or the release of energy. And then you need to link this to muscles as well. I need to explain that it's for contraction. If you say making or producing energy, you will not get any marks because it's against the laws of physics. You need to say it releases energy. Remember, you cannot create or destroy energy. Okay, the next one is still question A, and it's asking you to explain the link between rough endoplasmic reticulum and enzyme secreting cells from salivary glands. So we asked you to read lines 7 and 8. So read lines 7 and 8 and then make sure you explain that link. So the link is enzymes are proteins, and proteins are synthesized or they are made on ribosomes. Okay, remember ribosomes are on the side of rough endoplasmic reticulum. So let's have a look at the examiner's report and see how students did with that. Superficial answers. So muscles have mitochondria because we need muscles in everyday life will not gain you credit. Neither will a very vague understanding of the function of organelles. And you will lose a mark if you say that mitochondria make energy because they release it, otherwise you're against the laws of physics. You also need to make sure your ideas are correctly placed in terms of ATP produced. And remember, muscles do not respire. Respiration takes place in the mitochondria. Many candidates were, however, clear on the role of rough endoplasmic reticulum, but didn't refer to ribosomes. So when you're talking about RER, make sure you talk about ribosomes as well. Okay. So, question B. So this time is referring you against the passage. So use information in the passage to explain how a tadpole's tail is absorbed as a tadpole changes into a frog. Now again, this is an example of where they will take an organism that you haven't studied in depth and they will ask you a question on it to throw you. However, just because we've not done tadpoles in depth in college does not mean that you do not have the knowledge to answer this question. If you read the last paragraph, you will see that it talks about lysosomes in the cells of a tail. So you need to think about why the increase in lysosomes in the cell of a tail, and therefore you are using information from the passage as well, will mean that the tail can be absorbed. So pause video and have a go. It is because lysosomes produce or contain enzymes, and these break down or hydrolyze proteins and substances or cells of a tail. Okay, so it's that simple. Because the lysosomes have the hydrolytic enzymes, they break down the substances, the proteins, the cells of the tail. However, there was a bit of confusion here for students because they talked about lymphocytes and phagocytes. However, it's actually lysosomes that they're talking about. So they got confused with absorption by lymphocytes and phagocytes in the immune system, where actually it's lysosomes that are involved in digesting the tail. There's also responses about destroying the tail. It's not destroyed, it's digested. So do not be thrown by that. So, last one. Starting with some lettuce leaves, describe how you would obtain a sample of undamaged chloroplasts. Use your knowledge of cell fractionation and ultracentrifugation to answer this question. This is a question, is there something you might have done to obtain chlorophyll? So you might have done the first part of this when you're doing chromatography of chlorophyll. So have a think about that. It's 6 marks. So make sure that you have at least 6 steps in your process. You need to start with the lettuce leaves and you're going to describe your process. And it also says use your knowledge of cell fractionation and ultracentrifugation to answer this question. So make sure you refer to cell fractionation and ultracentrifugation. Okay? I'd give yourself 6 minutes to do this. Pause video and have a go. Okay, so you have up to 8 points that you can put in here. So the first step would be to chop up or any reference to breaking up the lettuce. Sometimes you might put it in a liquidizer. You'd say it's cold, you'd say it's in a buffer solution, and you'd say it's isotonic. Okay, so remember this is to preserve your chloroplasts and your cells. And needs to be in the same water potential so they do not get water being brought in by osmosis and bursting, or water leaves by osmosis and they shrivel up, liquorinate. You need to then filter and centrifuge filtrate. Okay, so filter your, make sure you get rid of any old lettuce stuff, and then centrifuge your filtrate. You then centrifuge your supernatant. Okay, so you take out your filtrate, just the supernatant bit, the liquid on top, and you use it at a higher speed, and you'll find chloroplasts in the second pellet. So once you've got rid of all the rubbish that you don't need, you take off that supernatant, do it at a higher speed, and you'll get your chloroplasts, which have been protected because they are cold, they're in that pH buffer, and it's at isotonic, same water potential. So let's see where students went wrong here. So responses to this part of the question were frequently marred by misuse of terminology. So homogenate and supernatation, fractionation, centrifugation were often confused. So make sure the terminology is on point when you sit the exam. Make sure you know what everything means. Now, where possible, your examiners ignored the incorrect uses. So they did try and give you a benefit of the doubt, but where it obscured your underlying meaning, marks were withheld. Most candidates appreciate the significance of adding a cold isotonic buffer, although this is generally added at a later stage in the procedure. So it's like one of the first things you do. Straight after you cut it up, it's in a cold isotonic buffer. From this point on, many accounts became less convincing. Homogenization as a process which broke open cells, perhaps not understood, with many answers giving the impression that whole leaves or small pieces of leaf were centrifuged. Homogenization will break up your cells. And few referred to filtering before centrifugation. There's no point trying to centrifuge something that's not been filtered, or you'll end up with just a load of plant stuff in the bottom. While others appeared to have lost track of precisely what they were centrifuging, supernatant or pellet. So be really clear with those steps when you're asked one of these questions. Okay, so they're the application questions. So you should now feel more confident answering these. You should be able to define different command words for a start. You should feel more comfortable with reading A-level exam questions, and you should be able to confidently answer your application questions.